Hey, I'm happy to introduce Dr. Jared here. Jared got his PhD in Indiana University in 2017, and since then, he started with a postdoc at Northwestern University at the Proteomics Center of Excellence with Neil Kelleher, where he joined the ranks of top-down mass spectrometrists. He also was one of the pioneers in individual ion mass spectrometry, so not just single cells, but single ions in Orbitraps. And today he'll be talking to us about some proteoform imaging mass spectrometry. Please welcome Jared.
I had uploaded my talk to the one computer. Don't know where that one went. Let's go. Oh, there we are. Great. Nope.
Right here. Oh, you set that up.
Oh, sure, thank you. Thank you. All right, thank you for the wonderful introduction. Today I'll be talking about probing the proteome of differentiating human skin keratinocytes using a relatively new technique within the Kelleher Research Group, single nuclei proteoform imaging. Before I jump into applications, I'd first like to talk about the underlying technology that allows these measurements to take place. As we all know, mass spectrometry doesn't actually measure the mass of our molecules, right? It measures the mass to charge ratio. When we think of an Orbitrap analyzer, as an ion oscillates around the central electrode, the frequency through a Fourier transform is related to the mass to charge ratio. Now, there's a subsection of mass spectrometry known as charge detection mass spectrometry. It's been around for a few decades, and it uses individual ions rather than an ensemble of ions to make a measurement. So we'll have singular ions that oscillate around the central electrode. Once again, we'll determine its M over Z through its frequency measurement. However, now we can look for these individual ions, the rate of integrated signal on the outer electrode of this Orbitrap analyzer. And we find that the signal induced is proportional to the charge of the ion. So now we can accurately measure not only the mass to charge ratio, but the charge of each individual ion, giving us its exact true mass. So you might be thinking, why is this important? If we have a complex sample, such as Kexel lysate, we don't do any separations with it. We do gel-free fractionation, though. We take the 0 to 25 kilodalton fraction, shoot into the mass spectrometer. We expect there to be thousands of proteoforms. On top of this, we get splitting of charge states here over the M over Z domain, and we get a really, really complex spectrum. It's so complex, we actually do not see any peaks. However, if we start to consolidate these various charge state distributions into one mass distribution, we go from no data to this very rich now mass spectrum. If you look at the x-axis down here, we're in the mass domain, kilodaltons. We're able to identify through a mass tag approach about 550 proteoforms with these really nice isotopic distributions that are able to match to theoretical distributions. So we'll now couple this technology with the differentiation of skin keratinocytes. We have a really wonderful collaborator in the dermatology department, Northwestern University, Xiaomin Bao. What she'll do is take discarded human skin tissue, isolate primary epidermal cells. I'll find that when she plates this, only the undifferentiated or progenitor states stick to the plate. And then to induce differentiation, it's kind of what we see here through the epidermis up until finally the cells are then shedded. And she'll do this with calcium chloride over the course of 4 days. We'll take these two different states, break the cells open, isolate the nuclei from the keratinocytes, and then we'll drop cast them on positively charged slides. Here's just a small picture of what a few of these single nuclei look like. They're about 5 microns in size. This varies from what the keratinocyte cells are, about 10 to 12 microns themselves. We'll then subject this to something we've been doing over the past few years, single cell proteoform imaging mass spectrometry, or SCPIMS. We'll use a nanodesi based approach pioneered by the Julia Laskin lab. We'll have a denatured solvent running through this first capillary here. It'll form a liquid-liquid bridge with the slide. As we raster that across the surface, it'll extract out proteoforms from the nuclei, or just depictions of a cell, into the secondary capillary. And then it'll ionize that into the mass spectrometer where we'll perform our individual ion analysis. And we have 3 different stages here before. This is the nanodesi probe, and this is a picture of the cell. We'll be doing this with nuclei though. Where we have before, on, and then after sampling. And we can see in the chromatogram here, we have a little bit of background before and after. However, we start extracting out these individual ions here when we're on top of the cell. We like to call this a cellogram. So to gain enough statistics for our various states, we'll sample 17,000 single nuclei. Half of this is in the undifferentiated state, and then the other half is in the differentiated state. Here's just a subsection of what these extraction profiles look like. Each one of these ticks is the sampling of a singular nuclei. So we have sampled 65, throughput of about 65 nuclei per hour. So it takes us about 11 days to collect these 17,000 data points. We find that for each one of these extraction events, we get about 6,500 individual ion signals they're able to use, and this corresponds to an isotope of an intact proteoform. Now, there's a lot we can do with this data. The first thing we'll do, though, is take everything aggregated together to be able to see what we have present. We are using intact mass tag approach. So we find that with across these 17,000 single nuclei, we'll have greater than 75 million charge-assigned ions here in this butterfly plot that we'll make. Undifferentiated is in red, differentiated is in black. And using our intact mass tag approach, we've previously taken a created a top-down database using the same sample set. And we have 265 high-confidence proteoforms we're able to determine through isotopic matching here. Here's just a few of these insets. You can see what some of these distributions look like. We get a lot of ribosomal binding proteins, a lot of histones, of course. And here's the H4 region, acetylations, methylations. Here's just another inset here of various proteoform landscapes of H2B and H2A. So now that we've determined kind of what we have within the entire data set, we can start breaking it back down, the puzzle back down into its different components. Shown here is just one of the nuclei that we sampled, number 1035. You can see what those 6,500 ion signals start to look like here in the mass domain for a singular nucleus that was sampled. We can start to see then how many ions go into each proteoform distribution. And then we also have a scoring system that we've invented here, proteoform assignment scoring, we call it PAS score. And this looks at, there's a few examples of what some of these various proteoforms look like for this single nucleus that we've sampled here. Do we get the relative abundance that we'd expect toward the center of this Gaussian isotopic distribution? And what's the accuracy of these individual ions to the actual individual isotopes of our theoretical distributions? That all goes into the scoring. Here on the left-hand side, you can see a rather high score. It scales from about 1, which is 100% confidence, down to 0, which is 0% confidence. You can see we populate for our single cell, our single nucleus here in black versus the theoretical distribution in purple. Here we populated pretty well. We got our nice Gaussian, almost like distribution, and our mass accuracy is really well, so we have a higher proteoform assignment score. And then on the right-hand side, you can see we populate more of the periphery here rather than the center, as we'd expect, and some of the accuracy is slightly off, so it has a lower proteoform assignment score. So we do this for all the proteoforms that we have for each nuclei. Then we could take the data set itself and we've started sequestering it to clustering. So we know what proteoforms go into each nuclei that we've sampled, and then performing clustering, we find in this example we have 5 distinct clusters we've determined. We find that these clusters are populated by both differentiated and undifferentiated nuclei. And so some of these clusters have about a 50-50 mix. Some of them are more populated by our differentiated nuclei, so for cluster 1. And then cluster 2, some of them are more populated by our undifferentiated nuclei that we've seen. And here it just breaks it down to components underneath. Now, there's a lot of data here, and to try to get an idea about what we should start to target, we did run a bottom-up histone panel to see what is upregulated and differentiated here in blue, and then upregulated and undifferentiated here in yellow. And just for the sake of time, I'll show just one example of what we've been able to determine. So we'll look at the H4K20 monomethyl. We can look at how this is expressed within these various clusters. And we find that here for cluster 4, for the monomethyl, it is highly expressed here in cluster 4 and lowly expressed here in cluster 2 and 3. And then if we move to the dimethyl, we find that it is more lowly expressed now here in cluster 4, higher expressed in cluster 0, 1, and 3, and then more lowly expressed still in cluster 2. We can look at the ions, for these specific nuclei and start to plot this back into their mass spectra to see how it corresponds with the plots on the right-hand side. And we find when we compare cluster 4 and cluster 3 for histone H4, the monomethyl is decreased as you go from cluster 4 to 3, as we can see on the right-hand side, but it's increased in relative abundance here in cluster 3 for the dimethyl. Similarly, we can do this once again for cluster 4 and 2, where we find that both the monomethyl as well as the dimethyl are both decreased in relative abundance here for cluster 2. We can also go back and look then at our bottom-up histone assay, see how this relates to some of the abundance that we find there. So the monomethyl here we find for our undifferentiated is more highly expressed. And then for our dimethyl, we find that although it's significantly more closer, differentiated is highly expressed, which we find that, and there's been some papers on this, that the monomethyl itself is corresponding with cell growth. And then as you move to the dimethyl and trimethyl, you get more gene silencing. So with that, I'd like to thank Northwestern Proteomics, of course, Neil Kelleher. We have a lot of postdocs, biostats folks that have started looking at this. This is kind of our first soiree into clustering and moving toward this style of analysis. So we still have a lot to learn. Thermo Fisher Scientific for all the individual ion work and development. A lot of our first single cell platform development was done in conjunction with the Sweedler group. And then, of course, for our skin sample preparation, Xiaomin Bao. So, all right, thank you. I'd be happy to take any questions.
Thank you very much for a great introduction to our single cell imaging. I'm curious, so of course histones are very abundant in most of these cells. Have you guys looked at different solvent mechanisms for potentially biasing to different parts of the cell, like, specifically membrane ones, more hydrophobic, or detergents in your solvents to kind of extract that information?
Yeah, so that's a wonderful question. We've really started thinking about coupling, and I know other groups have done this too, kind of the single cell and then single nuclei based sampling to kind of increase proteome coverage. Our depth is still not that deep. I mean, we're 265 proteoforms deep. We're not in the thousands. But yeah, we found that, changing, even denaturing solvent, adding more acid, you can bias different histones and modifications. So yeah, there's a lot to explore there. We just haven't done it yet. Yeah. Mm-hmm.
Any other questions?
If you manage to fish out a goldfish out of a pond, what would you ask it to give you in terms of improving the power of single ion imaging on the side of sample prep, instrumentation, data analytics? What would be your first wishes that the goldfish can grant you?
So you're talking about next steps then? Or
that's right, next steps. And sort of thinking really big picture, not being too concerned about resource. Let's say that Thermo and Bruker are willing to devote all of their R&D budget on the instrumentation side.
Right.
A little bit giving you the opportunity outside of practical constraints. What do you think are the biggest opportunities, promises that you would like to see realized?
Yeah, well, I mean, one of the biggest limitations that we have, of course, are databases that we use and matching. So intact mass tag approach, there's only so much you can do. We try to boost all of our identifications with taking the SAM data type and running top-down analysis or using previously determined bottom-up data sets, but having enough sensitivity to be able to do tandem mass spectrometry or something like that to actually have the sample and then try to analyze what exactly those proteoforms would be. Amazing, right, from what we do. So that's a sensitivity, and we just don't have enough ion counts to be able to do fragmentation, correspond that back. But I mean, that would be the dream, right?
Thank you.
Thank you. So in your 500 to 600 protoforms that you see in nuclei, how large those protoforms can be? Are you limited in the molecular mass of those protoforms? And if you funnel them down to proteins from protoforms, then what is the representation of the proteome? 100 proteins, 50 proteins? What? Give us, please, more detail sense.
Yeah, so for a lot of what we see, I mean, I showed a few examples. I mean, histones are highly modified, right? So the amount of proteins that we see, I mean, are probably, especially with this, less than 100. With cells, rather than just nuclei, it's higher. Sometimes we see just one proteoform per protein, depending. But yeah, as for the limit itself, I mean, we've shown using this style of analysis in a joint paper with Julia Laskin that we can go up to 70-ish kilodaltons. So we're a little bit higher. I know sometimes with the LCMS, top down, we're limited to maybe 30, 35 kilodaltons. So we can start to access a little bit higher. As we take these charge states and start to piece them together into one mass distribution, it really increases the signal to noise. So we can sample higher into that proteome. As for the depth, we just need to get, we usually have lower injection times for our analysis event, and we just need to be able to make it so that we can start sampling some of the more lower abundant proteoforms. We just aren't there yet. Yeah.
So what is your explanation for this limitation by molecular mass? So is it ion suppression? Is it like channeling into multiple charge states? How do you explain it?
Yep. So absolutely. So right, the larger it is, the more isotopic channel and charge state channels it has to fill. So for us to even vary that, you probably need a higher concentration to be able to see that, right? It's just too down in the weeds. The concentration that there is in the single cell just probably isn't high enough.
Can it be also ionization suppression when ionizable histones suppress everything else?
Yep. Yeah, absolutely.
Thank you.
So I think I saw on one of your slides that you estimate about 6,500 ions per nuclei is what you're observing. What is causing a limitation to that number, and how could it go up by like 100-fold? Because there's presumably more than 6,000 proteins in the nucleus.
Yeah, so especially for some of our top-down work, we're very sensitive to adduct salts, and that will also lead to ion suppression. So we've started increasing washing protocols and working through a lot of that. So we just need to get better at cleaning our sample, funneling all the ions directly into the mass spectrometer. It's probably just a transfer is what it is. It's just low abundance to begin with, and to transfer in and getting the ions out and up and in. It's just where we're currently limited. Yeah, there should be significantly more. Right, of course, proteins, proteoforms that are present. Right, in all the other talks we've seen, there's thousands. Right.
Great. Well, let's thank Jared again.